Hi everyone, today I'm going to be teaching you a little bit more about RNAi, or RNA interference. As mentioned previously, RNAi stands for RNA interference, and its discovery even won the scientist responsible the 2006 Nobel Prize. So this technology has been around for almost 20 years. RNAi is the process of using RNA to find and regulate specific genes in order to correct mistakes in the genetic code. Once RNAi is programmed to find the problem gene, it can also silence it. That silencing will definitely come back later in the presentation as we talk about limitations. In order to understand RNAi, first we have to understand the pathways of RNAi. So first, RNAi is triggered by double-stranded RNA, or dsRNA. During RNAi, long double-stranded RNA is cut into small fragments by an enzyme called dicer. These small fragments, called siRNAs, or small interfering RNAs, bind to proteins. These are often called argonauts proteins, and you can see that in the graphic on the other side of the slide. After binding to an argonate protein, one strand of the double-stranded RNA is removed, leaving the remaining strand available to bind to mRNA. Once the argonate protein is bound, it can cleave the mRNA, which would destroy it, or regulate the target sequence in other ways. Now, as with any experiment, RNAi also has some important controls. I'm going to talk about 5 different controls, but only 3 of them are going to be used in every RNAi experiment, or should be used in every RNAi experiment. The first of those is a positive control, which is used to establish the experimental setup. The second is a negative control, which should not have any homology to a known mammalian gene. And then we're going to skip down to the untransfected cell control, which should also be used in every RNAi experiment. And this is when you carry out gene expression analysis on cells that have not been treated. And then transfection control and mock transfection control will only be used in specific scenarios. So they are not as commonly used. And then I got all this information from Qiagen. So this is a company that provides many reagents and lots of what you would need if you were actually going to do an RNAi experiment. Before we get into clinical applications, it's important to discuss some limitations of RNAi. Most importantly, RNAi silencing methods have high off-target effects. This means that RNAi will sometimes silence unintended RNA targets, resulting in modified phenotypes, which can be a huge problem for gene screening experiments, as well as just the well-being of the study subjects. The off-target effect in RNAi can be one of 2 types, either sequence-independent or sequence-dependent. And in some experiments, the siRNAs trigger an interferon-activated pathway in a sequence-independent manner, which results in an increased expression of interferon-regulated genes. Sometimes that can be very problematic in genetic experiments. In contrast, CRISPR systems, which we studied in Module 1, they did initially have some sequence-specific off-target effects, but because CRISPR has been around for so long and because it is used so extensively, there are some CRISPR design tools now that have enabled finding guide RNAs that exhibit minimal off-target effects. Thus, comparative studies have shown that CRISPR has far fewer off-target effects than RNAi. Now, how can we use RNAi in real life? There are many clinical applications of RNAi, including the FDA-approved therapeutic patisiran. This drug is used to treat transthyretin amyloidosis, a life-threatening neurodegenerative disease, which is genetic and driven by the deposition of TTR amyloids primarily in the nervous system, heart, and gastrointestinal tract, although it can be present in other organ systems. The patisiran works by silencing both the wild type and mutant TTR mRNAs in hepatocytes, which are liver cells, to reduce the serum levels of the TTR protein. Now, to achieve broad silencing activity against TTR mRNA variants, the antisense strand targets the 3' untranslated region of the mRNA. Now, it's important to note that patisiran siRNA is not a fully modified, metabolically stable siRNA and does not have a targeting ligand for enhanced uptake by hepatocytes. Instead, patisiran is formulated for uptake by hepatocytes. Finally, let's talk about some ways that RNAi can be applied to our course MIP 611. First off, RNAi is essential for next generation sequencing, which we studied in Module 2, as both a manner of silencing certain genes and detecting problem genes, such as those that cause disease. As mentioned earlier, RNAi and CRISPR, which we learned about in Module 1, are both used for genetic experiments, and CRISPR is sometimes advantageous over RNAi due to fewer off-target effects. Protein expression and analysis, which is also included in this module, Module 4, is an essential component of understanding RNAi pathways, which we discussed in slide number 2. And as with most microbiological methods, they would not be possible without the foundational principles of Module 1, like the central dogma, vector selection, or PCR. Thank you so much for listening, and I hope you learned something about RNAi.